A warm Axiom welcome to you all. Good morning, good evening, everyone. Thank you for joining us today. As we are midway in 2021 and heading into ASCO, the Axiom think tank wanted to give you a quick preview of the latest scientific discoveries and potential clinical breakthroughs. It is a pure privilege to have Dr. Pat LoRusso join us today from Yale School of Medicine to give us her insights. Dr. LoRusso is an expert in early stage clinical trials.
So before we dive into the interesting findings for today, I'd like to provide a quick overview of what the next 45 minutes will hold. So we'll start off by looking at the breadth of early stage ADCs that were presented at AACR with a double click on Teliso-V clinical data and a new STN ADC from Seagen. Then we'll move on to some updates about KRAS resistance and the SHP2 pathway. And finally, we'll finish up with some astonishing efficacy from a couple early stage therapies. And the way that this session is going to work, similarly to some of our past town halls, at the end of each topic, we'll have a few minutes to discuss questions from the town hall audience. So whether you're joining us from YouTube or from LinkedIn Live, please go ahead and submit your questions through that forum. And then at the end of each of these topic sessions, we'll have time for that Q&A. So with that, I will turn things over to Dr. LoRusso to discuss the variety of updates from the land of antibody-drug conjugates.
Thank you very much, Anne. And I want to thank you all for joining me today, whether it's morning, afternoon, or evening, in whatever time zone you're joining me from. So I think AACR really had a lot of really interesting compounds this year. The beauty of AACR is that we get many times a preclinical portfolio, and which gives us preclinical insight into what we may expect clinically in the next few years, presented at major meetings and in clinical trials. And also there were some really good early phase clinical trials that were presented as well, both of which I'd like to just briefly expound upon today. So as you can see, there was a significant number of antibody-drug conjugates that were presented. Remember, most of your antibody-drug conjugates are monoclonal antibodies that have a unique payload, so that we're delivering not only a monoclonal antibody, which has the potential of efficacy, but also a payload which, by virtue of using an antibody-drug conjugate, can get into the tumor cell and kill the cell by a variety of different mechanisms predicated on the payload or the drug that is in the payload. So as you can see here, there were many different targets preclinically. Most of these antibody-drug conjugates were presented preclinically. There is one that we'll be talking about briefly that was also presented clinically. But you see that although there were a lot of different tumors that are being targeted with these antibody-drug conjugates, non-small cell lung cancer overall, with an emphasis on non-small cell lung cancer, is probably one of the most prevalent targets that the class of ADCs, at least preclinically, is investigating currently. Next slide. What we're going to be talking about today are two: the Seagen STNV compound, the whose target is sialothomsin-novu, which is a totally unique and novel target for an antibody-drug conjugate, as well as the telisotuzumab vedotin, which is targeted at c-Met and was a clinical study that was presented orally with a focus, obviously because of the target c-Met, on non-small cell lung cancer. Next slide. So this is the first drug that is that was presented preclinically, the Seattle Genetics drug targeting sialothomsin-novu. And what you see, which is really quite an interesting and novel target, is that this target is really expressed or overexpressed on tumor cells, but it is not really demonstrated. There's not real demonstration on normal cells. So theoretically, it has the advantage of having a wide spectrum of efficacy because it can target many of the tumor cells. And hopefully, we don't know this until we really get into the clinic, but hopefully it will be very selective targeting tumor with sparing of normal cells, thereby enhancing efficacy with minimum toxicity. And being a novel target, it may have quite a significant impact if it pans out clinically as it did preclinically. And what you see here on the right is a waterfall plot. And actually, each of these lines represents a different PDX histologic tumor subtype, which showed that in a lot of these preclinical in vivo models, and as you can see by the color coding, they primarily looked at gastric, ovarian, esophageal, and non-small cell lung cancer. There was significant efficacy, predominantly, as you can see, in the ovarian and non-small cell lines. So the phase 1 clinical trial is currently recruiting, and hopefully we'll see that data maybe at next year's AACR. Next slide. And then the clinical trial that I wanted to just briefly touch on was the antibody drug conjugate that targets c-Met. It was a single drug study, and this was a very interesting trial design. The compound is Teliso-V, or telisotuzumab vedotin, and basically, as you see here, the trial, I think, was the first stage of the trial, at least, was very, very well designed, looking at specific subsets of tumors, for instance, non-squamous EGFR wild type, EGFR mutant, and then squamous cancers. And then they further subdivided these tumor types, or these subgroups, into c-Met high versus c-Met intermediate. And I think by subcategorizing, or by breaking this recruitment into subgroups and then additional subgroups, it led to, I think, very interesting data. And understandably so, because we don't know if all of these tumor types and subgroups based on EGFR wild type or mutant going into this trial would have equal efficacy, and it could have skewed the overall outcome into a totally negative trial. But what you see on the right in terms of efficacy endpoints was that the overall response rate in EGFR wild type patients was 35% versus only 13% in the EGFR mutant patient population. And if you further subcategorized it into their second subgroup, into c-Met high versus c-Met intermediate, you see that in the wild type, there's almost a 54% overall response rate in the c-Met high, with only about an 18% overall response rate in the EGFR mutant c-Met high. And then if you looked at the intermediate, there was about a 25% response rate in the EGFR wild type. But very interestingly, no EGFR mutant patient that only had c-Met intermediate had a response. Now, keep in mind that the numbers are very small in each of these subgroups, but nonetheless, it is food for thought. And the greater response rates that were seen in the EGFR wild type c-Met high subgroup met enough statistical significance that they are moving on to a phase 2 clinical trial in that subset of patients. So it's going to be extremely interesting to eventually see what the outcome of that study is, as well as the combination study with an EGFR targeted agent. I'm sorry, Anne.
No, I was going to ask a clarifying question here. So the idea of having a greater response rate among c-Met high versus c-Met intermediate patients makes a lot of sense based on sort of the mechanism of action of an ADC and looking to combine with an EGFR in future. But why, what is your thinking, or maybe a hypothesis around why they would have seen such a significantly stronger response rate in the EGFR wild type patients overall compared to those EGFR mutant? Is it just the different biology of those tumor types?
Well, I think it's multifactorial, first of all. I think biology factors in, but specifically related to the biology, you now have two drivers. You have two oncogenic drivers. You have EGFR driving the tumor as well as MET. And they're obviously somewhat exclusive in terms of how they impact on tumor growth. And I think that that's why it's going to be extremely important. One of the trials that is either planned or ongoing is going to be in combination, I believe, with osimertinib or another EGFR targeted drug. So that, I think, if we can circumvent toxicity of the two drug combination, it's going to be extremely important to determine whether or not using a drug that targets both MET and a drug that targets both EGFR and combining them together will we be able to see at least a good of response in the c-Met high, if not better, than what we saw with the monoclonal in the EGFR wild type? And will we be able to capture then some additional response or greater response versus like an EGFR targeted drug only when we combine both the MET and the EGFR targeted drug in patients that only had c-Met intermediate expression or amplification? Next. So this is, I think, very interesting. Although we don't have, and it's very difficult to look at, a PFS comparator as an example when you don't have standard of care control arms. So we don't really have PFS data in this small study. And also the numbers were probably way too small to do that. But I think the two graphs that you see here, both the waterfall plot as well as the swimmer's plot, are very telling. And this is in the EGFR wild type, as you can see. But what you see is at least you see a clinical benefit in probably 80% of patients with this waterfall plot on the left. And what you also see is that you see a lot of the patients remained on study for a very significant period of time. And in fact, if you looked specifically at these numbers, the duration of response for the c-Met high group that were EGFR wild type was about 6.9 months median, which is quite good. And the overall response, which I think is very telling, and these patients stayed on study for a long time. So again, I'm excited about looking at a randomized phase 2/3, as well as a combination study in the EGFR mutant population.
Yeah, very promising. And the longest patients in the study, I think, are approaching 1 year on therapy, which is very, very exciting for this patient population.
Right. And these patients had to have at least 2 lines of prior treatment in order to go on this study. So you're talking about a heavily pretreated patient population. And they did get, this was quite a bit of therapeutic benefit in such a heavily pretreated population.
So now we have time for a couple of questions here. We've had a few questions come in. If you're watching from either YouTube or LinkedIn Live, go ahead and send those questions through. I might start us off with a question about the STN antibody from Seagen. So one of the interesting things about this drug, if I'm not mistaken, is that the epitope isn't just a specific protein. It's a glycosylated epitope that's found on multiple different proteins. So Dr. LoRusso, what do you think that means sort of functionally? Does that make it a more valuable target from an ADC perspective?
Yeah, I do think so, because I think this is a novel approach as well, because most of the ADCs, have monoclonal antibodies that are just targeting the protein. But if this can target a glycosylated protein, I think it enhances the probability of efficacy versus toxicity. And I think that is going to be key. And also because this glycosylated protein is manifest on multiple different tumor types, I think that there's possibility of a wide spectrum of efficacy as well with this agent.
Mm-hmm. And the issue of toxicity, I mean, you alluded to the goal of combining the c-Met therapy with an EGFR treatment. How do you, if you were going to give guidance to someone thinking about building a clinical trial or even enrolling a patient in a clinical trial, how do you think about evaluating the safety versus the potential efficacy benefit once you start combining multiple sort of targeted innovative therapies here?
Yeah, so one of the important things when you're thinking about it is what is the monotherapy toxicities and how do they overlap in terms of toxicity profile?
Okay.
So I think we may be able to get away with this combination in this patient population because of the somewhat different toxicity profile, or at least the predominant toxicities. But nonetheless, when you start combining these drugs, you do have to at least do a phase 1 run-in so that you can assure that the safety is there before you go full blast into your phase 2 trial with the drug combination. So obviously, you're going to have to identify which of those 2 drugs is going to be your predominant driver and which are you going to try to maximize when you initially start your combination and then escalate from there. But good point. You have to watch that.
And so what that would look like in practice, would that mean if both drugs have hematological toxicities, they probably are a poorer option for combination than if maybe one has primarily hematological toxicities and another has liver toxicities or dermatological issues?
Absolutely, absolutely. I mean, if you look at immune checkpoint inhibitors in combination, so, for instance, most of your immune checkpoint inhibitors, when you're giving them in combination with another drug or chemotherapy, you can usually go full dose of both chemotherapy and checkpoint inhibitors because the overlap in toxicity is minimal. The toxicity profiles are quite distinct. But you're right, when you have two drugs that have similar toxicities like hematologic, you're not going to probably be able to get away with full dose. And you're also potentially going to have to work towards schedule alteration. I think we're going to be okay here with the CMET and the osimertinib combinations that are being planned or ongoing.
Perfect. And one last question on CMET coming from the audience. CMET as a target is not necessarily brand new. This is something that's been evaluated in the past. There was a late monoclonal antibody that failed in late phases. How is this approach different? Is there an advantage to taking the ADC strategy to be able to target CMET in some of these patient populations?
Yeah, I think there is, because it allows you to deliver to the cell the payload, the bendoitin in this case, which is going to get intracellularly because of the amplification of MET. That antibody drug conjugate will be targeted towards that cell and will then be taken up intracellularly and be able to give you greater efficacy because of that. So with something like a CMET antibody drug conjugate, we did see some preliminary efficacy with CMET inhibitors in the past. Obviously, there's been issues. They've been somewhat slow to develop, but this is not only taking advantage of targeting with the monoclonal antibody and potentially getting efficacy with the monoclonal antibody, but also taking advantage of the backbone, the bendoitin as well, to be able to give you a twofold bang for your buck, so to speak. And I mean, there have been other examples, even with drugs like HER3 targeted agents, and we saw that data last year presented. We've tried, many of us have done many monoclonal antibody studies targeting HER3 with no success. But now they put a backbone that is, the topo-1 targeted agent from Daiichi as an example, and a drug, a monoclonal that really didn't have much efficacy, now being able to deliver that backbone intracellularly has led to really quite profound results. So, but the advantage of this compound is you can target CMET as well as deliver the cytotoxic agent, which I believe a drug like the AbbVie compound telisotuzumab vedotin is giving you a twofold effect.
And that's so exciting, Dr. LoRusso, given the first slide that we opened up here. I think we listed a dozen ADCs in preclinical development, and that wasn't even all of the ones that were presented at the study. So if this technology really is bringing in this, hit them twice, twofold effect in terms of efficacy, it's very exciting to see all of the new technology that's coming up and hopefully bringing some efficacy to these very hard to treat patient populations.
Right. The challenge, I think, will be as we develop these compounds to develop also novel backbones, because many of the tumors have already seen, as an example, mitotic poisons. And so the future, I think, is not only the beauty of these novel antibody drug conjugates with novel targets, but also the development of novel backbones, not novel cytotoxics or backbones that can give you that double whammy effect. Thank you.
Yep. And that's something we'll be looking for, right, in all of the coming congresses.
Absolutely.
More data on that.
Absolutely.
All right, so we'd like to move on then, thank you, to the next section, next set of topics that we have here, which is around this idea of undruggable targets or undruggable pathways. This is a sort of a quick summary slide. There were a lot of moving pieces related to the KRAS story at the recent AACR. We've sort of bucket them into three pieces here. One being resistance, patients who are receiving KRAS inhibitors, but eventually develop resistance to those tumors, learning about who those patients are, what kind of mutations they have that are giving them that type of resistance. A couple of presentations on related pathways. So we're going to talk here about SHP2 in particular, and then introduction and some first ever data from a number of very early stage KRAS inhibitors coming to join the party. So this is back to you, Dr. LoRusso, for the first paper here on resistance mechanisms.
Yeah, so, being able to drug KRAS has been just an amazing discovery. I mean, Shaukat and colleagues, and then the companies that have been moving forward to not only initially target KRAS, but now to build on that targeting. The Amgen 510 compound that was presented last year and the year before demonstrated that, yes, we can target KRAS. Specifically, that drug was for KRAS G12C. But I think what we're going to be seeing in the next couple of years is targeting yet other KRAS targets, such as KRAS G12D. But in terms of targeting KRAS G12C, what we also saw, especially with the Amgen compound, is that monotherapy, we got efficacy. But, that efficacy was durable for a period of time. But eventually, many of the patients, the majority of the patients, went on to subsequently progress. So the Dana-Farber team presented this beautiful study of looking at acquired resistance to KRAS G12C inhibition in solid tumors, looking at various mechanisms. So these were patients that had some type of sequencing or assessment of their baseline tumor, and then they were given a KRAS G12C compound, and then at progression, they looked at their tumor either by biopsying the tumor itself or looking at circulating tumor DNA or both. They had 18 patients that they assessed, 12 of which, there were 18 patients of the 30 or so that they studied that they could not identify a known mechanism of resistance. So 60% of the patients, they could not, with their current tools and with the samples that they had, identify resistance mechanisms, but they were able to do so in about 40% of patients, or 12 of them. And what they identified was that they could bucket the resistance mechanisms into three buckets. Those that had acquired KRAS alterations, very similar to what we've seen in the past, as an example with EGFR. And so these patients, when they progressed, they had new KRAS alterations. The second bucket was patients' tumors that had developed acquired alterations in the RTK/MAP kinase pathway. And then finally, the third bucket that they were able to identify were tumors that developed acquired gene fusions that were not present at baseline. Of the latter group, the gene fusions, what was very interesting, but keep in mind that this is a very, very small number of patients. They only have 12 here. The acquired gene fusions were only identified in the colorectal population. But what I think is amazing and what's going to be challenging is the variability in only 12 patients of mechanisms of resistance. The challenge is going to be, how do we tackle this to prevent or delay acquired resistance in terms of combinations, sequencing, etc.? And where do we begin to treat to be able to prevent this from happening? And what happens? How do we treat these patients after they have developed these different mechanisms of resistance? And will we be able to bucket our therapeutic technologies and techniques similarly, or will these separate buckets lead us to separate potential pathways of therapeutic intervention? Next slide. So basically, one of the drugs that was presented that Anne told you about was one of the targets, which is SHP2. And as you can see here, SHP2 is part of the upstream cellular machinery that transduces receptor tyrosine kinase activity into activated KRAS. It helps facilitate the activation of KRAS as well as other RTK-dependent signaling cascades. And preclinical data has shown, and we'll be looking at that in a few minutes, that if you combine SHP2 inhibitors with KRAS inhibitors, it strengthens the anti-tumor activity or enhances it, and may delay or prevent the development of resistance. The mechanisms of this enhanced anti-tumor activity are not fully understood and could be accounted for by one or both of the following mechanisms. As you can see, either impeding the tumor's ability to develop resistance by hitting upstream of the target, particularly an upstream target in the RTK pathway, or a deeper response due to interfering with the same target at two separate points. Next slide. So basically, this data was published, I believe, a year ago, but I thought it was really nice data, not presented at AACR this year, obviously. But what it did show, and this is preclinically, looking at ERK phosphorylation, as well as in vivo combinations of a SHP2 inhibitor with a KRAS G12C inhibitor, the Mirati compound that's currently in early phase clinical trials. And what it showed was that there's significant alteration of ERK phosphorylation with the combination, as you can see on the left in this in vitro study, with the combination versus monotherapy, regardless of how high you go. And it does seem to be dose proportional with monotherapy KRAS G12C inhibitor, but that ERK phosphorylation is essentially the same regardless of the dose of the KRAS G12C inhibitor. Very, very potent phosphorylation in vitro. And that data did pan out in vivo, as you can see on the right. Looking at two separate tumor models, what you identify is significant inhibition with the combination, which is in green, versus the monotherapy arms, as you can see there, either blue or red. And even with the second arm, even with the second model, the H358, what you see is that the combination and the monotherapy of the initial Mirati compound were pretty much superimposed during the dosing interval, but the dotted line represents discontinuation of dosing in these in vivo models. And what you see is recovery of tumor growth with monotherapy. But when you had given the combination, it significantly continues to alter tumor regrowth for an additional 50 to 60 days. Quite profound. Hopefully we'll see this when we go into this in vivo drug combination into man. Next slide. So basically, this was the SHP2 inhibitor that was presented by Revolution. And what you can see here is this was a phase 1 trial. None of these patients were given a KRAS G12C inhibitor prior to going on to this study, but many of these patients were heavily pretreated. And what you do see is that there is monotherapy activity with this agent. And it's primarily in tumors that were either G12C/D or G12V. There was no activity really seen in patients whose tumors harbored a G12S or a G12A mutation. But nonetheless, with the G12C compounds out there, as well as G12D compounds that will be hopefully moving forward soon into the clinic, there's a lot of promise potentially of this drug in combination with those agents, provided that the toxicity is not profound with the combination over monotherapy. But I think that this phase 1 trial, although we didn't see earth-shattering efficacy, but we did see some efficacy, and you got to keep in mind that there were a lot of different allelic alterations as well as a lot of different dose levels. I think it validates SHP2 as a target and supports the development of the combination therapies. Next slide.
Also, to Dr. LoRusso, to follow up on some of the things you mentioned before we move to this next slide, I want to ask you a question.
Yes.
Of this agent, do you think that there's a role for SHP2 inhibition before, like in combination with KRAS inhibitors, like you said, but also after disease progression on KRAS inhibitors?
Yeah, I think that's a very interesting question. I think potentially yes. But, we're going to have to see that pan out. And I think that's a very, very, interesting concept, especially trying to incorporate that into some type of a clinical trial. The question I have is, do you then, if you're going to treat patients who've progressed on KRAS inhibitors, do you then treat with a monotherapy SHP2 inhibitor, or do you keep the KRAS inhibitor on board and add the SHP2 inhibitor?
Yeah, I think it's an even interesting question. I think it also depends on the type of resistance mutations in this context and how patients progress, whether that's either through RTK alterations or other genomic events. Maybe some patients might be more sensitive to this type of inhibition in the progressive state.
Yeah, and I think it lends itself to, do we serially sequence patients? Because do I think this combination would work, or SHP2 would work in a patient that's developed a fusion? Probably not.
Right.
But resistance by virtue of RTK? Possibly yes. And resistance by virtue of another KRAS allelic alteration? This data right here shows that not all KRAS allelic alterations are equally effective, with this combination, or even have any efficacy. Again, pointing here, your acquired KRAS alterations, possibly not, but your RTK MAP kinase alterations, possibly yes. Again, the time has come that we are probably, with some of these select agents, especially with some of these select molecular alterations that have many different resistance mechanisms, we're probably going to have to resequence these patients if we are going to think of, in the future especially, the best treatment path forward.
Thank you, Dr. LoRusso.
Yes. So the other things that were brought up, there are other SHP2 inhibitors that are now in combination with KRAS G12C inhibitors. And there have been multiple partnerships. Many of these SHP2 inhibitors have been developed by smaller biotech or companies that have gone into collaboration with either companies that are large or small that have the KRAS inhibitors. So you've got a large company like Amgen that has partnered with Revolution, Mirati, a smaller company that has partnered with Novartis, Genentech that has partnered with Relay, and obviously we all know Novartis. But because of this early preclinical data that's been seen both in vitro as well as in vivo in multiple different models, and the exciting clinical data of not only the KRAS inhibitors, but now the SHP2 inhibitors, many companies are moving forward. But again, just like you brought out previously, all that is acquired resistance is not created equal. So if you're just generically going forward, my concern is that you will not necessarily identify the significant value of these combinations. Again, there were many other small molecule inhibitors that were unveiled at AACR, primarily preclinically. I guess the question we could ask ourselves is, do we need more KRAS G12C inhibitors? What is the market for these agents now that we've got at least 3 or 4 that are in the clinic already? I think each of these actually has a specific added value. For instance, the Lilly compound is highly selective inhibitor with a predicted target occupancy greater than 90%. As you can see, significantly supersedes at least the target occupancy of the 2 most developed compounds, the Amgen compound as well as the Mirati compound. And maybe with this added target occupancy, we might see monotherapy complete responses. We didn't see that initially with several hundred patients treated with some of these other compounds. So that may be an added value of enhanced target occupancy. I think the Revolution compound is very, very interesting because it's targeting KRAS, but it's targeting it by a totally novel mechanism of action. It actually binds to the mutant KRAS in the on state, as opposed to the other inhibitors that target on the off state. And then finally, the Boehringer-Ingelheim compound is also highly selective, and it has significantly greater potency, at least in vivo in preclinical models. The question will be, does this potency equate to added efficacy clinically, or will it translate to increased toxicity? I think that is the question that will only be answered when we finally get this drug into the clinic. Next slide.
And Revolution Medicine's coming in with a big AACR, right? The first preclinical data here for their KRAS as well as the SHP2 inhibitor. So they had a lot of data in this story from that one relatively small group.
I know, very, very exciting. I mean, very small group, but they're working on a pathway and they're trying to really dissect it and understand it and try to tackle it head on. Yep. Kudos for Revolution.
A couple of questions coming in through the chats. First one being about KRAS combinations. Now that we're understanding some of the resistance mechanisms, does this suggest any sort of the way that combinations should be thought about in order to prevent resistance? And maybe thinking specifically around other targeted therapies versus combining with something like a chemotherapy.
Yeah, so, combining with the chemotherapy, a lot of these targeted agents, regardless of KRAS or anything else, have never really been, have never really panned out with combination chemotherapy. The hope is that we can get combination targeted therapies. I think one of the challenges, if indeed that small number of patients that Dana-Farber sequenced, if the data from that small number of patients pans out clinically, how do you predict what the mechanism of resistance is going to be for a specific tumor type? There were 12 patients in that group, and except for the fusions, I mean, there were a lot of different alterations in those 12 patients. So how do you predict in advance what combinations are going to be your best moving forward? So for instance, if you do give a SHP2 inhibitor as an example to try to maximize the RTK inhibition, will that increase more rapidly the different KRAS allelic alterations that we saw with some of the resistance mechanisms? That's the question that we have to answer, and I think we're only going to be able to answer that probably in the clinic, where we are seeing so much variability in mechanism of resistance.
Is this something where liquid biopsy or ongoing ctDNA monitoring might be able to play a role to avoid, having to rebiopsy some of these patients?
Yeah, I think that's a very good question. So, with the data that was presented from the Dana-Farber group, I think there were only 3 patients that had paired biopsies. A lot of them were identified by ctDNA. And I think many years ago, David Hyman presented a really good study where he was testing a novel drug. And basically, he followed that patient's ctDNA. And when he started seeing that there was an increased alteration of a different pathway that would have led to resistance, he added a second drug onto that patient. So, ctDNA is, 5 years ago, it wasn't, it wasn't as widely used and as well developed as it is now, but perhaps the future of drug development is going to be either starting with these monotherapies, and when we start seeing a signal of resistance, adding a second drug onto that patient's monotherapy in hopes of delaying the development of tumor growth. I think you're very right. Tools like ctDNA may be the future of how we develop drugs and how we treat patients, more importantly.
And I think that actually leads nicely into our third section here, which is around some of these very targeted therapies, at least for the FGFR story. So FGFR is an interesting marker where it doesn't affect the majority of patients in any of these tumor types. But for everything listed here, something like 5 to 10%, a little bit more in urethral, do have FGFR alterations, whether that is amplifications or mutations or rearrangements. And for those patients, those rare patients where they do have FGFR, does seem to be a really key driver here. So I think there was a paper here on cholangiocarcinoma Dr. LoRusso, do you want to talk us through what they found for cholangiocarcinoma?
Yes. Can we go back to the previous slides? We're going to be focusing on a drug that was presented at AACR, futibatinib. And as you see here, as Ann just pointed out, FGFR is not seen in many different tumors. I mean, it's seen in many different tumors, but in a very, very small percentage of patients. But what she also talked about was the different mechanisms of FGFR expression and how it manifests. And basically, go to the next slide. What was beautiful about this study is many FGFR inhibitors have failed in the past. They failed many times because we've been targeting tumors that have had FGFR amplification or expression. But what they did with the development of futibatinib, they only went into one tumor type, intrahepatic cholangiocarcinoma, which is a very aggressive tumor, but does have FGFR fusions to a significant degree. They're found in about 14% of intrahepatic cholangiocarcinomas. And so rather than going after amplification and expression, which have not really panned out in terms of developing previous generations of drugs, what they did with this drug is they took it and they looked at one tumor type that has FGFR fusions. And they treated patients regardless of what their number of prior regimens were. And in fact, as you can see here, the majority of patients that they treated were patients that had had at least 3 prior treatments for their intrahepatic cholangiocarcinoma. And what they saw, which I think is really amazing, is they saw significant responses across the board. Overall, they saw about a 42% response rate. But that response rate was not predicated on the number of prior treatments that these patients had. In other words, those patients that were less heavily pretreated did not do better than those that were more heavily pretreated, which was amazing. For these patients that have a very highly aggressive tumor, this is pretty profound data. And as you can see here, looking at the waterfall plot in these FGFR FGFR fusion iCCA patients, across the board, there was phenomenal response. And as you can see from the dotted line, that's where the PR to CR would break, at least 30%. There was a clinical benefit in at least 80% of these patients. And what was also beautiful is that the duration of response median was 9.7 months. Many of these people after third-line therapy would not really be living, but maybe maximum a few months. So this drug has significant benefit in these patients. And there's one patient at the time of data cutoff for reporting at AACR that was on study for at least 2 years. This reminds me of the TRK fusion story, that hitting the TRK fusion was really astronomical. And in the TRK scenario, we saw responses regardless of histologic subtype. Now, the question is here, we are seeing these beautiful responses in intrahepatic cholangiocarcinoma, even multiple line therapy patients. Will this data translate to other histologic tumor types that also harbor fusions? That's the million-dollar question. And hopefully with this impressive data and moving this drug forward, we'll start looking at that patient population as well. Not a common population of patients. Not a lot of tumors have that FGFR fusion, but a significant enough of them that developing this drug across the board, if there's benefit, would be very important. Kind of again like the TRK fusion story. Not a lot of TRK fusion tumors out there, but it has proven a very beneficial drug in the fusion population patients. Next slide. So the other drug and the last drug that we want to talk about is the WEE1 inhibitor that was presented.
I was going to say, Dr. LoRusso, if you mind, I'd like to provide an overview for this, given that WEE1 is
perfect. Please do so. I apologize, Lee.
Right, no problem. So when we're speaking about WEE1 in this context of the story, we're going to demonstrate some or show some data that a WEE1 targeted agent demonstrated some efficacy in patients that were heavily pretreated. But in this context, we're going to talk about WEE1 as a target. And so WEE1 is involved in DNA damage response. And as you can see on the left-hand side, this diagram shows that upon DNA damage, ATR and other checkpoint kinases activate WEE1 to essentially halt the cell cycle so that tumor cells can repair that DNA damage that takes place. And so inhibiting WEE1 in this context essentially drives those cells to replicate even with that damaged DNA, and there's an anti-tumor effect associated with that.
Beautiful overview. Thank you so very much. So basically, we didn't, what was really presented with this compound, and this is the Zentalis WEE1 inhibitor, they did present the toxicity data of the phase 1 trial. Primarily nausea and diarrhea were the two most prevalent toxicities. But they went on to show some vignettes, and I think they had 5 vignettes all total. But I think what's impressive is looking specifically at the second vignette in the middle. This was a patient with metastatic ovarian cancer who had had 11 prior lines of therapy in the metastatic setting. And she had a tumor reduction of 56%. Pretty amazing. Even the metastatic colorectal patient who had had 5 lines of prior therapy had a 42% tumor reduction. So this drug, I think, has demonstrated efficacy in monotherapy. And I think the exciting future with this compound is, will it be continued to be developed as monotherapy, or are we going to be looking at this drug in the future in drug combinations? One last thing. There is rumor that this drug can cross the blood-brain barrier. So interesting whether or not we're going to be looking at this drug in the future in patients that have tumors driven by HRD that have brain metastasis, or to delay the onset of brain metastasis in high-risk patients.
That's actually something you mentioned just now would want to follow up on. You mentioned patients with HRD. What are the types of patients we need to be looking for therapy such as WEE1? Are these going to be patients with, BRCA mutations or unique genomic alterations? Like, what does the ideal patient population look like for targeting, WEE1 in this context?
Well, what they are looking at is they are looking at homologous recombination defects such as BRCA, such as ATR, especially ATR as an example. The verdict is out about ATM or CHEK or PALB2, but those are also interesting molecular signatures that we need to think about as we're developing a WEE1 inhibitor and advancing it forward.
Great. So that is about bringing us to the end of our session today. We want to thank you, Dr. LoRusso, for taking the time to speak with our participants and our audience today. Before we sign off, I want to give a quick preview of some of the topics coming up here at ASCO. 1 month starts 1 month from tomorrow. Lot of data here, but I want to just point out a couple of things. We're going to see the readout of the Olympia trial, so looking at a PARP inhibitor in early breast cancer. Continued progress from checkpoint inhibitors, both in new tumors and combinations, as well as moving forward up to the adjuvant setting. A couple of similar themes to AACR that we've just talked about in terms of updates from ADCs and some exciting new data from KRAS in lung cancer. And then some other new and promising therapies, including readout from the VISION trial of radioligand therapy in hormone-resistant prostate cancer. So we are looking forward to seeing these data come out over the next month. Once ASCO is done, we'll be hosting a number of town halls. So mark your calendars to join us in talking to the clinical experts about how the data we see at ASCO will change practice. So with that, a final thank you to Dr. LoRusso. Thank you to our participants, and we will see you next time.
Thank you all for joining. Have a good week.
Thank you so much.